Welcome, welcome, welcome to summer 2021 Stanford Bio webinar series number 6. Today is an extremely important day, I think, and actually one of the most fun days, at least for what I'm teaching. I think it is the most fun day for what I'm teaching because we're going to go over DNA logic. So everything you've been learning up to this point has kind of been foundational. This part's, I think, actually thinking like an engineer. You're actually thinking to problem solve, you're thinking in advanced ways, you're thinking almost like more like electrical engineering, more like an engineer. And I think that's really cool and kind of the bread and butter of synthetic biology, which I know at here at Stanford we love, right? Bioengineering is pretty much focused on synthetic biology. So that's super exciting. So yeah, we'll be doing DNA logic. Woo, woo. Again, feel free to unmute, ask questions in the chat or whatever. And I'm super excited. This is my favorite subject that we're gonna teach. Okay. So I want to start with a little overview map here of what you're seeing. And we can kind of think of this as a cell. We can also think of this as a black box, if you will. So you have some sort of input, some stuff goes in, you have an output. And I want to kind of divide this box up into 3 sections. And you'll be here, I'll be using these terms interchangeably, one with the biological terms that I'll show you in a second what this looks like, but also with more of the logic terms. And so we have 3 parts. We have sensors, we have controllers, and we have actuators. You can think of sensors as inputs and actuators as outputs, and controllers are kind of the logic in between. I'll show you what this looks like on a plasmid in a second. But for the inputs, you can see inputs could be anything from chemicals, it could be light, it could just be some sort of energy input, it could be chemicals from the dirt, depending what it is in, it could be nucleic acids, it could be RNA. There's all sorts different types of inputs that you can't have to really activate this. The point of a sensor is it senses these inputs. It waits for these inputs in order to then activate what the controllers do. Again, the controllers are internal logic of your circuits. They'll kind of do the bread and butter and also have the, I guess, encode the steps or encode what it takes to produce your outputs, which are the actuators. And there's a bunch of different outputs. You could have proteins, just general proteins. You could have proteins that are markers like GFP and RFP. You could have proteins that produce chemicals. You can be really creative with all this stuff. Honestly, it's endless. Honestly, I think it's kind of endless. These are puzzle pieces that you can put together in any way you want as an engineer. Just be creative. These are the basic parts. We're going to really focus more on the sensors and the controllers, which is labeled internal logic circuits here today. The actuators is more protein engineering, protein modeling. I know, I think Alan may talk a little bit more about that in his enzymology lectures that are coming later, but I really want to focus on sensors and internal logic today. Great, awesome. We'll continue. Okay, so I wanted to bring back the kind of basic plasmid structure we've been looking at in the last couple of webinars, just so you can really understand where these kind of input into. So you can see the sensors are about the promoter region. The actuators are your output, so that's going to be your gene of interest. And then transcription and translation, so things in between, maybe the amount of RBS or the type of RBS you have, or other kind of steps, or maybe combination of plasmids would be your controllers, and these are going to be kind of your logic, is a way to think of it. And then of course, the origin of replication selection markers, these are separate. These are more for experimental design. So yeah, this is how it fits in a plasmid, pretty basic, but I think it's nice to see. So you can connect it to previous lectures. Great. I thought I had a question there. Does it ever miss the question, Alan?
Not for me.
Oh, I exit. Oh, I exit. I swear I had a question. Oh, I did it. I'm so silly. Okay, never mind. I just don't know my slides that well. All right, so now I want to talk about feedback loops. We'll start. So I want to talk about positive feedback loops first. And I think a really good example, and I'm sure maybe you've seen this before because y'all are cattle farmers, I know, is cattle's in panic. Okay, so I want you to close your eyes and imagine with me. A bunch of cattle are panicking, or 3 cattle start panicking. And then the ones next to them also start panicking. And you got 2 cows next to each cow. So you can do the math there. You now have 9 cows panicking, and then 2 cows next to them. So now you have 27 cows panicking, and so on and so forth. And so you can see how this would kind of increase on itself and feed itself in a way that would kind of exponentially grow, as you see in this graph here. And so this is a positive feedback loop. And of course, this graph isn't perfect. You'd probably, maybe you have 180 cows on your farm, and so this will level off at some point. You can only have 180 cows panicking. But that's an example of a positive feedback. And these are just very basic examples. I just want us to understand the logic before we go forward. So I thought that was a fun example. All right, now we want the opposite. We want a negative feedback. And so here's a negative feedback. And we can try to maybe apply the cow example to it too. So let's say that you're a farmer, you wanted to, your cows would panic just because they have anxiety. And so you wanted to force feed them like Xanax every time they panicked. And so now they start panicking, and then you activate it, your Xanax shoots, and they eat the Xanax up. That calms them down for a bit. And then they panic, then they get the Xanax, then you calm them down. That's a negative feedback loop, and you can see that here. B would activate A. This would be B is the panic, activates A, your Xanax, which would suppress B. Think of it like that. And oftentimes these have an equilibrium point to them. And so you can get your graph here, which you would have this kind of oscillation, and the degree and the size of these, I guess, oscillations really depend on your system and the context that it's in. And then it kind of reach this equilibrium point. So maybe the cows only have so much energy, and so they go to bed. That'd be an example. Obviously a very realistic example, and so that's why I shared it. So let's continue. Okay, it worked. So I wanted you guys to try to be creative too and think of some feedback loops. Maybe these are some you imagined yourself, or maybe there's some you already know that exist. What are some feedback loops? Oh, and we got a chat too. I do love cows. They're so friendly. And here's the thing too, and while we wait for some answers, cows, since they're domesticated, they act just like dogs. I don't know if you know that. Like they'll come up to you and you can pet them. They want to get in your house and things like that, but you can't let them because they're stinky little monsters. But I love them. Insulin and glucose levels. Yeah. Okay, now something to add. Is that positive or negative feedback? I want you to add that in there too. Would that be positive or negative feedback? Whoever put that in, or if someone else wants to answer that. But that's a great example. That's a very classic example. Yeah, very good. Yeah, I can, see that one. Yeah. There's some other ones. Very good. Yeah, it is a negative feedback for sure. Yeah, what are some other ones? All right, let's get creative here. I love thinking about these. Oftentimes I'll like pick something I'll look at, like I'll think of grass and maybe the dirt it's in. I'll try to think of all the loops and all the kind of logic that goes into growing grass or water, or how those things, like water on grass, could be a feedback loop in some sort of way. I think there's a lot of people on ice cream, and that's a positive feedback loop for sure. For sure. Yeah, that's a really good one. That's a really good one. Yeah, body temperature. Yeah, these are awesome. A lot of things that deal with the endocrine system are negative feedback loops. So downstream of certain production of hormones, you will produce one hormone that will then go upstream and turn off the other hormone productions. Fruit ripening, greenhouse gas effects. Sure, that's a positive feedback loop. Both of those, it's really good. Fruit ripening is a really awesome one. If you put certain fruit under black light, you can kind of see that in action, which is, I think, really cool. But yeah, awesome. Okay, we had fun with that, and now it's time to get serious. So, oh no, how do you do this? Oh, and I got it. So I had an example too, and I know you guys are very, very fond of your cytochrome BD reductases, because we all are. Me and Alan were searching up what the hell that meant earlier today. So don't worry. So I had an example of it. This is a synthetic biology example. The paper is, oh, I didn't link the paper. I'll send you all the paper at some point. But here's the example. And here's actually the feedback loop system. And so you have this constitutive promoter, which just means always on, a promoter that's on. And it makes this activator, LuxR. LuxR prime, it's a specific example, like it's a modified protein. And so this activator will then activate P-LuxI, which is your promoter LuxI, that will make more LuxR. And so now you have more LuxR that then goes back and activates more PLuxI, so on and so forth. And downstream of this, you have another gene of interest, which is your cytochrome BD reductase. And so this is so cytochrome BD reductase is used to decrease oxidative stress in bacteria. And so this can help them stay alive. And so this is an engineered example of a positive feedback loop. And so I have a question for you. So we have this downstream gene of interest. Why would a positive feedback loop as a tool be useful? What type of proteins do you think this could be useful for? When would you need a positive feedback loop? If you want to think about the graphs. Thank you. Thank you, Alan. So what types of proteins could you use positive feedback loops to make? I guess that's kind of a weird question. I guess, what are some attributes about the proteins? Think about that exponential graph. It's kind of a hard question. I think I'm asking it a little weird. Any answers? If not, I'll just kind of. Yeah, that could be an example. Yeah. I guess I'm asking. So oftentimes, so for example, yeah, that could be an example. I guess I was asking a little bit of a different question. I asked it poorly, my bad. But a lot of cell wall proteins are lowly expressed, and oftentimes when we try to get them out, you can't get a lot of them. And so if you really want to increase the expression of your gene of interest or your protein of interest, you'd use a positive feedback loop. This is, I guess, more of kind of what I was getting at. But those are other, in the chat, great examples of when you would actually maybe see this. And I can show you maybe later examples too, where you kind of see a positive feedback. But does that make sense? If you really want to increase expression of certain proteins, you're going to get that exponential curve. I want you to think of it qualitatively, and you're going to get a lot of that protein. And so maybe there's not a ton of them. But here's a great example, very simple thing. So yeah, great. Any questions about that? And if I'm talking too fast, I realize I do that. There's a button on Zoom that you can say slow down because I go 500 miles a minute. And every time I try to slow down, I do not. But great. And then I want to give an example of just a basic, very generic negative feedback. I couldn't find a very simple model like I did for the positive feedback on my Google searches. So here's my example of my negative feedback. And so okay, you have your promoter, you have your gene of interest Y, and this gets produced. It gets then translated. And once it's translated, protein Y works as a repressor. And so this will stop then the transcription of your gene Y. And this is an example of a negative feedback, more of a synthetic biology way of thinking about it. And so, yeah, you can see how these can be super powerful. And in fact, I have a question coming up. So why would we want to have negative feedback loops? How could these be useful as tools for synthetic biology? Or even naturally, they do occur naturally, right? Stopping cancer, sure. Think more maintaining homeostasis, very good. That was kind of the homeostasis is a very important word when we think about that. What are some other reasons you can think of? But yeah, homeostasis is kind of the word I was looking for. So great job. You guys are so smart. Yeah. Think of them more as like protein expression. Why would you want negative feedback loops for protein expression? Or expression of certain, genes. Like, what does it mean to have homeostasis for these proteins? Yeah, exactly. Right? Good, exactly, yeah. And sure, I want you to remember qualitatively what this looks like. You saw the graph where it oscillates a little bit until it reaches kind of that set point of equilibrium, which is homeostasis. And so we want to control the amount of expression of these proteins until we can get it so it kind of reaches its equilibrium point. Good, awesome. So that's the power of positive feedback loops and negative feedback loops. And again, this is just kind of more of a overhead. So when we start actually dealing with maybe some of these systems, you're comfortable with the logic behind them. But yeah, you're all doing great. Okay, let's continue. So promoter region again is when I want you to think of promoters, I want you to think of sensors. because often upstream of promoters, you'll have some sort of sensor when you're trying to design these systems. So we're going to talk about sensors now. And I have another question because I was full of them, I guess. All right, I want you to put on your little engineering caps or hard hats. Ding, ding. What is a switch? What is a switch? Very generically, if I'm an alien and on off, yeah, on FF. What is a switch? Alan's a chemistry major. He needs to hear some of this stuff. He doesn't. Just kidding. Yeah, got you. Moo. That's funny. Any ideas of what a switch is? There's really, I'm not trying to trick you. I'm not trying to trick you. I just want you to think about like, what is the logic behind a switch? If I walk into a room, what do I need to do? You know, if it's dark and I can't see, what is a switch? Oh, they're not appearing? Ah, that's so sad. I was like, damn, stumped y'all. Well, I guess things pull everywhere. I guess write them in the chat and I can read them out loud. I don't know. Okay, so tool to change something from positive to negative or vice versa is a good answer. Something that can be flipped to an on or off state. Yeah, activate or suppress something else. On, off, again, expressing a certain action, then nullifying it. Enable, disable an action. Yeah, y'all got it. You know, that's pretty much exactly what I was looking for. On, off. Now, I guess we can also feel free to unmute or put in the chat. Why might this be powerful? Why would we want to turn something on, off? Why would we want to maybe program this? How can this be useful in everyday life or even, control gene expression? Yeah, exactly. And so I want us to think a lot about sensors as we're going to start thinking of them as somewhat as an on-off switch. Collin switches to become a chem major. That's not happening. I can tell you that one. Too high of an activation energy for that one. Give us more control of effect. Yeah, good. Yeah, great answers. And pull everywhere is canceled. We're never using it again. Okay. So I wanted to go over kind of a basic example of this. So we have some transcription factor that binds to an operator. And when it does so, it kind of turns this promoter sequence on. You have some ligand chemical that binds then to this. You have a conformational change of this protein. So the protein changes shapes in a way that it can no longer bind to the operator, which then turns the promoter off. So this switch is off, on, or on, off. How does this switch work? If once I add the ligand I, chemical I, on, off, or off, on.
Collin, can you clarify what you mean by that question?
Yeah, so I guess in the normal state, what is this protein? And then once I add chemical I, or what is this, I guess, promoter sequence? Then once I add chemical I, what is it after? So initial state to latter state after adding chemical I. On, off. Yeah, very good. It's on, off. And that's kind of really the skinny of it, right? And this is extremely powerful when you think about it or activating it for all the reasons you said previously. And this is kind of a generic example. We'll kind of look at how this is used a little bit further. Now, I want to kind of show you so once I turn this promoter region on, off, so you're going to have a decrease in your gene of interest. However, I want you to notice maybe something interesting a little bit about this graph is the fact that it's what we call sigmoid. So it has this kind of S, I guess it's like an inverse sigmoid because usually sigmoids increase, but whatever. It's got this nice S curve to it. And usually with a toggle or with an on-off switch, what you'll see is kind of a plateau. If I could draw that for you, maybe I can. Oh, maybe this is a waste of time, but we'll see. What you'd see is on, off, right? This being on, and then this being off, right? But we don't see that. And why might not we see that? What are some reasons maybe that we don't see this kind of steep on-off? It's gradual. Yeah, what's gradual? It is very gradual. Yeah, the slope is for sure more gradual. And again, if you're reading this graph, this is expression levels on the y-axis, so expression levels of your gene of interest. So maybe we're measuring fluorescence of GFP based on how much I is in there. Yeah, yeah, the expression is gradual, or almost the D expression, yeah. Yeah, Rohan, that's definitely right too. It's somewhat random, right? Not all I is going to bind to my transcription factor, causing conformational change, and so it's going to then cause a gradual change. And Alan's going to talk more about this when he gets more into the specific protein stuff. But yeah, there's a lot of reasons to it. This is kind of compounding factors. And a lot of biophysics, once you guys get to that, is kind of about this, trying to model the randomness that happens when things have to bind to, like ligands have to bind to receptors and things like that. Yeah, analog versus dialogue. Yeah, that's a good way of thinking it too. Awesome. You guys are thinking great. I love this. This is awesome. So great. Okay, and we're going to continue. And so I want to then flip the switch on you and start with an on to or off to on, right? And so here's an example of that. And so we have some transcription factor that acts as a repressor. Listen, I thought that joke was so funny. And when something binds to it, it then conformationally changes this transcription factor, so the protein will change in a way that it can no longer bind to the operator region. And this allows for the promoter to then be bound to or whatever and activates the gene of interest. So this is off on. What do you imagine so again, I have inducer. So let's say that's I. My y-axis is gene of expression, or yeah, gene of interest. What do you imagine that graph looks like? So how would it be different than the last graph we looked at? Qualitatively, what would we mind expression levels do as we increase the amount of inducer, or this little star here look like? Flipped horizontally. Yeah, exactly. And so this is a true sigmoid function. And so for the similar reasons as you said previously, you're going to have, off and then on. And you have this one inflection point kind of here. And then why might you notice, as you notice, this isn't just exponential, like we may see with a positive feedback loop, but this is flat, plateauing at some point. Why do you think it's kind of coming to a stop? What's going on? Biologically. Reach an equilibrium. Yeah. What are some other reasons too? Or places to bind. Yeah. Yep. Yeah. You guys are kind of getting to the point. Awesome. Great. Okay. So we'll continue. But yeah, so that's your kind of basic examples of these on-off switches in your promoter regions. I want to give an example that's really often used. If you ever work in a synthetic biology lab, you're going to use IPTG. IPTG is a synthetic version of allolactose, which is a sugar, oops, being the sugar suffix. And so you have this promoter C, sub C, which is constitutive promoter, again, means it's just on, that produces LAC-I, which is the inducer protein. This inducer protein will go and bind to the LAC-O, which is the LAC operator, and turn the circuit off, I guess I could say. Now I add this inducer. Again, this is my synthetic allolactose, which is IPTG. This will then bind to the LAC-I protein, which again is a repressor protein. Conformational change, so it will no longer bind to this operator region and have this on. So this is an off to on switch once I add IPTG, if you want to look at it from that framework. Again, I show you this because this is used all the time. And I'm sure if you continue your pursuits in bioengineering or work in synthetic biology labs, you'll have IPTG. And you can see why this is powerful, right? This gives you as an engineer a control of whether or not your gene of interest is ever expressed, which is, you can, for obvious reasons, what you want, because you want to control things. You want to have your experiments under as much control as possible. Great. Let's continue. I wanted to give another example, and this one's found often in eukaryotes. And so what's going on here? What's kind of going on here when you look at this? So we have promoter that produces this TET-on third generation gene of interest, which is this TET-on third generation protein, which is a common, this is a common, again, very common in eukaryotes to do this, kind of like the IPTG equivalent. And this, you want to, express your gene of interest. The way you do this is you dope in doxycycline. Doxycycline will then bind to this 3G protein, causing some conformational change. This will bind to the promoter region, allowing it to then be expressed. You're kind of getting the logic here. I'm going to go over this again and again because it's super important to really get this in your bones. This is your goal and the different ways to do this. But it works pretty much the same way to IPTG. But this is a eukaryotic example. I thought it would be good to show. Some other eukaryotic examples you can think of, and I think maybe Alan, if you can find a paper on it, would be the estrogen induction. I think that's a really common one, and I think there's a lot of cool projects right now on synthetically producing estrogen and other hormones. And so people are trying to, as we speak, are working on these kind of circuits to produce these things, and this is an integral part to them. And of course, they have different proteins that work as different chemicals that work as inducers and different proteins that work as activators. So very cool, very cool. Okay, so now we're going to talk about controllers. And so I had to put my boy from Canada, Drake, on there. And controllers, again, when you look at controllers, we talked about sensors just previously. Sensors are kind of, you associate with your promoter region. Controllers are more of the internal logic. If we look at our black box system, if you can remember from the first slide, and these work as kind of the logic to figure out transcription translation rates almost, or what steps you need in order to transcribe, translate, what rules you need to follow, so on and so forth. And I have some examples to show you in order to really make this make sense. So let's get into it. So first I wanted to show you some Boolean logic. And if any of you have coded before or looked at schematics, maybe you've seen some of this. But if you've not, I'm going to go over some of the basic ones. The one we're going to focus on a lot today is this AND gate. So AND gate, so let's say we have two binary values being 0 or 1. And an AND gate requires you to have both be on, so both need to be 1. And so you can see if X would be your outcome, of course, here. So if B or A are off, both off, you're not going to have it. If one's off, one's on, you're not going to have it. One's on, one's off, not going to have it. Both have to be on for an AND gate to then produce output X. Not is just the opposite. So if 0, then you have an output. 1, you don't have an output. Not AND, so anything that isn't AND. So you can, this would look logically tilde and ampersand. And that's kind of what that is. Or, so if either one is a on, then you're going to produce something. If both are off, then you won't produce something. That's how that works. Neither or neither, kind of. So if both are off, then you produce something. If one of them's on, you won't produce anything. Yeah. Then you have XOR, and this means only one of them is on. Then you'll produce something. And then XNOR, either they're all on or none of them are on, and that produces something. Otherwise, you will produce nothing, and X being your output. So these are very, these are your logic gates, and you're going to start seeing, you're going to see AND mostly. I think AND's the only one I use for later slides. But feel free to look more into these. It's called Boolean logic again. Thanks, Alan, for putting the estrogen induction in the chat. And wrapping your heads around these, you can get really creative with your circuit designs and your DNA device designs on how you can maybe put them together in order to produce some of these functions. If I had more time and this class got a little deeper, we could get more into this because I think there's some really cool ways to think about these and use them. But alas, we don't. If you have any questions and you want to go deeper into this, I can send you some great resources. Just email me. But awesome. All right, let's continue. Have all these gates been built in geogenic circuits? I'm sure. I'm sure they have. If you remember the paper from last time, or I have a Nature paper I can send. Wait, I'm going to send it really quick because I think it's a great paper. If I can find it. I think I have it. Biocomputation. Yeah, this is the cool biocomputation. Oh, wait. I want to see if I can find this paper real quick. Do I have it? Yeah. Check this paper out. This will kind of go into a lot of them that we see. And so, yeah, these have been put into all types of circuits, or a lot of these logic gates have been put into gene circuits. And you got to be creative to, I think, include some of them, but they're pretty cool. They're pretty cool. Do they all exist naturally? That's a good question. I'm not sure. It's something to look into. Oftentimes, though, when we think about these, we're thinking about them as synthetic biologists. So this whole section is as synthetic biologists. So again, that means kind of man-made biology. So how are we manipulating biology and how are we manipulating our plasmids in order to do something like this? Is how at least I want you to think about it. So we're thinking about these as engineers, but they may exist naturally. You always learn from the environment, always learn from nature. Island, do you know if they all exist naturally? I don't have a
I
great
don't know
answer.
for this example specifically, but I mean, for the image on screen right now, for example, you have a chaperone, you have a transcription factor. Maybe that exists naturally, but then maybe we put the promoter in front of our gene of interest, right? That's where we come in. And so, these things might be derived from natural sources, but yeah, we can still change them. It's a very common theme you might see throughout our presentations. But
yeah. So I guess the logic behind it is more man-made, but that's a great question. Okay, so I want to show you a simple 2-input AND gate, and then maybe we'll get a little more complex if we're all feeling confident. So input 1 would be some sort of plasmid that would produce the chaperone protein, and input 2 would be something that produces a transcription factor that requires this chaperone to help it bend in its 3D structure in the proper way to then bind to this later promoter that would then induce our output or activate our output. And so you can see input 1, this would be like A or X, I guess, in the example of this chart I have here. And input 2 would be B. Both of them need to be on, and we could turn these on maybe constitutively, so they're always 1. Or maybe we have something specific about the environment that would need to then turn them on. And so only getting our output when both X and Y, input 1 and input 2, are on. Now, of course, you can make this way, way, way more complicated. And so I want to show you a 4 input AND gate. So this would be like a double AND gated system that has 1 AND gate at the end. Just think of it maybe like a pyramid if we kind of put it like that, or I guess that, rotated that way. And so you can see that we have these systems of chaperones with their transcription factors going on. And so these would produce IPG chaperone with certain transcription factors that would then bind to this here. And this would be input 1 and 2. Input 2 and 4 would do the same thing. These need to come together in order to bind to this promoter sequence. This would then go on to produce these two, the chaperone with this transcription factor that then needs to bind to this. And so if you want to extend it, you'd have ABCD that then produces X, and you'd need to have 1, 1, 1, 1. All of these would need to be on, and then these would need to be on too. So just expanding it, and you can see how complex these can get, and they really do get complex. If you remember the paper Alan sent from last week from the Smolke lab, there is some really, really serious gene circuitry going on there, and that's even in eukaryote. That was in yeast, right, Alan? So that stuff, you get complicated. You get complicated. You have to deal with organelles and specific locations of the cell. Whew. You got to be creative. And so this is just kind of expanding how we can think about this and adding double AND gates in a way. And then our gene of interest here is RFP, which is red fluorescent protein. And so that would be kind of a marker, which is you fluoresce red. That means we can then tell and measure that it's on pretty easily. Just, think of experimental design when you have that. And this would be, I don't get into it, but again, your actuator or your output, this would be more actuator output design. What do you want to output? Why do you want to output it? If you just want to test to see if this system you have set up works, period, and you don't want to output any protein that does anything, outputting RFP or GFP would make sense, right? Because you just want to get some sort of fluorescence. Great. Okay, so I want to.
Out of curiosity, how do you know how leaky these are? Like the system with 4 inputs. If there's one leak, that might.
Well, with 4 inputs, I would imagine the leakiness kind of decreases because you'd have to have each one be leaky to X degree, right? Like each one's leaky for like, let's say 10% of the time, right? For all of them, you do 0.1 times 0.1 times 0.1 would be 0.1 to the 4th, right? And that's actually something that there's a good paper, and maybe if I can find it, was on ribosome binding leakiness, in which they chose different ribosome binding sites, and they like put a bunch of them together in order to decrease the amount of leakiness or to really control translation rates, which is kind of interesting. But yeah, the leakiness of these systems, I think oftentimes are negligible. I think it's when you have your simpler systems that they kind of you have to be careful and have to really watch out for it. This is also something that people do test for in lab, and often is very important. You want to be able to control and understand why you're getting the expression you're getting, and so you'd have to run some control experiments. If I don't induce these whatsoever, I don't add IPTG whatsoever, and I'm still getting RFP, then that's extremely leaky. Like that's a problem. And so you'd have to test for these things. Does that answer your question?
Yeah, that's really helpful. And I guess for those who don't know, it's like if you entered a room with the light switches off but the lights on. That's what leaky means, right? It's just leaky expression. Mm-hmm.
Yeah. So just get something that's not supposed to happen for random reasons. And this happens in biology a lot. We're dealing with cells that we don't control. Like we're engineering cells, but cells, they are a life form of their own and more complex than how we want to engineer them. And that's why there's the Build a Cell project, which is really cool. If you haven't looked into it, look into it. Yeah. Great. Okay, so I want to give an example of an AND gate that was used actually in this paper, Kempton et al. Alec, if you could, or Alan, sorry, if you could link that one. And so this one is trying to mark breast cancer genes. And so we can look at this AND gate here. So this is a 2-input AND gate system. And the first input is RRM2P, which is activating this promoter sequence. This is ribonucleotide reductase, which is a enzyme that works to make ribonucleotides into deoxyribonucleotides, which you can see this has been shown to be extremely active in cancerous cells because they're splitting all the time. They need a lot of DNA. And then the second one is like hypoxia. Cancer often is found in hypoxic environments, I guess is that term. And so this would activate this transcription factor that would then bind and activate this gene. Now you're going to ask what this gene is, and this is an example again of actuator design, if you want to think about it. And this is the N and C terminus of Cas12. And so that's a great question, Emil. I'll answer in a second. And so when these come together, then these have their 2 subunits of the Cas12 system that would then come together spontaneously and work to then edit the genome to produce therapeutic genes. And so you can see some of these here. And this would then help us to, as you can see, very powerful to mark breast cancer cells. By producing these genes, then we could tell that this is a breast cancer cell specifically, and based on the environment that is kind of unique to a cancerous cell. And in fact, if you don't believe me, here's a graph from the paper, and you can see the AND gating going on here. You should only see high expression when you have it 1, 1. So negative would be 0, positive would be 1. And so in breast cancer cells, if these would be your promoter sequences. If they're not on, both of them are on, you don't really get any significant expression. And in non-cancerous cells, you see no expression, even if they're both on. Or both there, sorry. Both there. These would be if they're both there, and then cancerous cells, you see that the environments do actually do so. And this would be what leakiness is, right? You do get some when just, because again, this is just those transcription factors. These are part of the cells natively. And so you get some leakiness there because there are some binding to the hypoxia operator sites. And then there's already these transcription factors present in the cell. And so you get some leakiness once those genes are induced, even though this is not even in the cell. So it's interesting. Yes, that makes sense. Great. So someone asked, what does DNA logic have in common with synthetic cells? So DNA logic, I guess synthetic cells, I'll talk a little bit later about cell therapy actually. and how we do this. But synthetic cells are more of the idea of, right, like you need DNA logic to build it, right? These are building parts, I guess, building blocks, right? Synthetic cells is just a control factor. Like if we can build our own cells, then we have control over these entire systems, and we can really engineer everything specifically about them. I think if they really don't have, like, they don't overlap in that sense, but one, you need one or the other, or these are both part of bioengineering, or two different fields of kind of bioengineering. And then I'll go into example of synthetic cells and how you actually use certain DNA logic in order to engineer cells. I think if that answers your question, let me know if I'm just kind of talking past it. But yeah, great, awesome. Okay, so I wanted to talk about this. This is a generational paper. You know, this is going to be, you know, this is already 21 years old. It's older than a lot of you, or all of you, besides me. Let's go. And so this paper is super important because it designed this little sequence thing. And so let's look at what this is doing. So we have promoter A. And so promoter A is on. You have a negative feedback loop, right? It will produce your gene of interest A that then goes and represses promoter B, which then you wouldn't produce gene B. If you have promoter B on, gene of interest B gets produced and goes back and represses promoter A. So A is not produced. Very important. Very important. What could this be used for? Let's get some, what do you think this could be used for? Or what is this? What could this be used for? This is a feedback loop. Yeah. But I guess this this design of your gene circuit in general, what is this useful for? Cyclic protein expression? No, I'm kind of getting something more general, like engineering wise. Like how could this be powerful? How could we use this as engineers? Think of maybe computationally, maybe like how hardware is. This is more of like a hardware design in a way. It's a toughie. Okay, so again, if A is on, B is off. If B is on, A is off, for sure. And so it works like a toggle. And so what you can use these for, I put a little Minecraft toggle there too. Thought that was good. You would understand certain states, right, are true about this. And so if I had a whole system of this design, I could test for certain states and I could keep some sort of memory, or over time have some sort of memory. Yeah, a little flip-flopping. Yeah, for sure. And so I put a picture here because it's kind of like a transistor. It's either on or off, and I could know what the cell's been through based on what gene's been on or off. And so in that sense, it's a memory. And hopefully you can see how that can be extremely useful in a lot of different experiments if I want to test in their different environmental states and things of that sort. So that's a landmark study and a landmark design. Okay, so let's try to look at this one. So we have, don't worry about the names. We have A, B, and C, and promoter A, promoter B, and promoter C. So what's happening here? So as A gets produced, it goes and reduces B. As B gets produced, it goes and reduces C, the amount of expression of C. And as C gets produced, it goes and reduces the amount of expression of A. So what is this an example of? It's kind of taking the system, adding one more to it. Now we have 3 in here. What is this an example of? So so let's say we had a lot of A. We'd have a lot less of B and a lot less of C. Right, but if we have a lot less of B, then C gets a lot, and then as C increases, then we'd have less of A. But if we have less of A, then B increases, and then we have less of C. You see how this is working? Kind of like a oscillator. What? So this is an oscillator design, and this is also pretty landmark, and it was published in the same year, I think, also in Nature. And you can, again, appreciate how oscillating designs may be important for, just having as a tool set. So extremely powerful tool, and you can oscillate. And so I hope that makes sense. Again, we can go through it. When Rohan was talking about earlier was the oscillation, I think, but that's what this is. So you need 3 for oscillation, 2 is just a toggle. Yeah, no worries, no worries. That's awesome. So yeah, so going through it again, just really quick. A turns off B, turns on, and then B being on turns off C. So if A is on, you turn off B, but then you produce a lot of C that turns on A, that produces a lot of B that turn off C, and so on and so forth. And so you can get some sort of cycle of these proteins being on and off, which is really powerful stuff. So now we're getting computations. Okay, so I want to talk about cell therapy a little bit too, and kind of how we can almost introduce this into making synthetic cells, which I think is really powerful. So we have chimeric antigen receptors, or these are called CAR-T. CAR-T cell therapy is used for leukemia, so it's used for certain type of blood cancers. And what it is you're engineering your T cells here to have these antigen receptors. These antigen receptors are chimeric because they do 2 things at once. They, one, bind to antigens on the cell surfaces of your tumors, and then also induce some sort of, once bound, induce some sort of reaction in the T cell that should kill the tumor cells, some cytotoxic behavior. This is super cool. This is kind of the forefront of cell therapy. A lot of medical companies are really getting into this, and it's really powerful. And so I guess we can talk about the steps on how this works. And you can kind of see maybe on using this as, how would you design this? Think about it in your head. So you remove blood from the patient to get their T cells. Then you insert your gene. This gene would need to produce your chimeric antigen receptor, which will have 2 subunits. One that actually induces, once activated, would activate some sort of T cell response, cytotoxic response. And then one that will also bind to the antigen on the tumor cell surface. We use leukemia because those are really well documented. You need to know what the antigen looks like really well, have really good structures for it in order to create the receptors, right? And so those are really well documented for leukemia, and it's also they're easy to access because it's a blood cancer. You then produce millions of these T cells and do a transfusion. You put them back into the patient, and all of a sudden they're working. This is FDA approved. There's a bunch of other cell therapies that are working to be approved too, but this is kind of like the forefront of technology, so I want to share this with you. And this is how I think the overlap of maybe some logic, right, creating these DNA structures to actually have a useful. So this is what you could think about too. How would you do this? So very, very cool stuff. I think that's all I had. I went so fast.
I do want to plug one more thing as well. So, Collin today has talked a lot about, different processes and different regulation systems. And it's all very qualitative, right? So like this turns on, this turns off, this turns on, this turns off. But then you might imagine, how do you actually model these mathematically and quantitatively? And that's where differential equations comes in. So I know you're all in high school, but if you have a chance to take differential equations, or once you're in college to take these DiffEq classes, do. They're super, super helpful. And if you want to think about how to actually mathematically model these changes, that's the language of differential equations in calculus.
Yeah. Differential equation. Yeah, modeling is all these differential equations. It's crazy. I'm in differential equations right now for my class. It's a hoot and a holler. It's actually really cool, but it can be tough. There's a great question in the chat on, so synthetic cells have applications. So yeah. These cells they're engineered, right? In some way synthetic. They're not completely synthetic, again, because we're getting them from a donor. Usually you get them from the person, but sometimes you have someone else donate the T cells. And so they're not like, we're not making the T cells from scratch. We don't have that power yet, but one day maybe. We're just engineering them in order to produce these specific antigen receptors, which are CAR receptors. And so this is CAR-T cell therapy. And it's really, really powerful. And go and look at other companies that are kind of doing this. There's some startups that are in this area. Cell therapy is really cool. But yeah, Alan's definitely right. I want to go back here, even like just quantifying these curves. Alan will talk about a little bit, I think later, but just understanding how to like quantify, like, okay, what the steepness of this. There's this thing I think called the Hill function, right, Alan? Yeah, that gets into more quantifying, like even how you express this. And because science, right, you have to quantify it. You can't just be all qualitative. And so even quantifying these logic systems and these expression levels, we have systems for that and stuff. And so I did the qualitative just because I think math's hard and I don't like it. I do like it, but it's not my favorite. But get into that in the nitty gritty, and you can actually get a really good fundamental understanding of what's happening here if you understand it. Mathematical modeling, super, super important. Covert Lab, which is a lab I've worked for in the past, Marcus Covert, who's a professor at Stanford, his biggest paper is whole cell modeling. And so he's modeled an entire entire E. coli, just computationally. It does almost all the functions that an E. coli could do, and it's completely modeled. And those systems are based off of differential equations of what Alan was talking about a second ago. And you can see why this is important because modeling is cheaper and faster. Using just like computational power to test these things, or even that's one way of doing them too, when you talk about modeling in the abstract. But then having to mathematically understand it, not qualitatively, but quantitatively understand what's happening or how much gene levels you're getting or why you're getting those, that's also very important to model. Yeah, and that's kind of biophysics is a lot about mathematically modeling. Like, for example, let's say I had, instead of just one region here being bound to, I had 3 regions here. There's error rates and stuff, and there's a lot of things that go into that. How would you mathematically model that to understand like what would be a likely expression of it and whether or not your system was meeting that or not? Yeah. And yeah, to understand that further, Alan will talk about it too. And so mathematics and biology, even though biology is kind of seen as not a very mathy science, I think bioengineering for sure is. You have to understand some differential equations, probably. You have to understand something, especially once you get to like higher level. Not in my classes even yet, but like grad level classes, you'll get it. What are some challenges with DNA logic? Oh, that's a great question. Leakiness is a good example of like a very basic challenge with it. But ensuring this, like some of these happen, right? Like look at all the steps that can go wrong here. And even so, I'll go to this example actually here. So when we produce, can leakiness be fixed? Yeah, there's some ways to fix it, depending. Again, like I said, like ribosome binding sites can become like more stringent is a way of doing that. Or changing some things about your promoter, maybe using a different promoter so it's not, a little more stringent, or you need more of it or whatever, right? Because these all have kind of rates to the level they express naturally. But another big example is just like ensuring these proteins do bind together. So example would be with Cas12. You can design these systems and they're designed in ways that theoretically work and then for some reason just don't. Either the proteins don't bind, they don't get translated or transcribed correctly. And it kind of becomes hard to really understand like why or why not that's happening. That's another example of where, these systems can kind of fall apart. And a problem with synthetic biology in general, especially when you get, this is just a 2-input system. Again, if you look at the paper from last week, the Smolky paper, that does some crazy stuff. And just that system working has like so many intricate pieces to it, tens and tens of different circuits in it going all at once or needing to rely on each other. Any one of them fails, it's kind of game over, right? And so you can see some of the issues with that, especially as we get more complex with this stuff. So yeah, that's a great question.
Yeah, and these circuits, you can make them and you can engineer them. But understanding how these circuits are in natural systems, that's a whole topic in itself as well, right? Like biochemistry, a lot of it deals with regulation, right? How do you actually control how much enzyme there is, how much protein there is? All of that is circuitry. It's this, but it evolved naturally. So it's kind of cool.
Yeah. And all of these are fixable in a way, right? There's rates to this stuff and error rates and stuff like that you have to understand are going to come with the territory. If you, again, look at this, right? These are error rates. That's a great question. I think there's a how are parts obtained / characterized. So do you mean like parts of the plasmid, or like you're talking about like promoter sequences? Gene of interest, is that what you mean? Or like, what do you mean by parts? Okay, cool. Yeah, some of them are, we talked about a little bit how they're characterized in the past, right? Sometimes that's a bioinformatics challenge. Sometimes that's a crystallization challenge. A lot of those are computational challenges. And then the parts, a lot of these parts are already stored online. If you remember from the benchling lecture, which isn't on YouTube yet, but it will be. We have databases for a lot of these parts, T7, all that stuff. And so you can go and find these parts online for a lot of them, and oftentimes they're used. Or a lot of them are sourced directly from nature. There's a lot of like really cool animals out there that have like the whole caste system that was found in a bacteria and then engineered later, right? So a lot of these parts are found naturally, and then we engineer them and then put them online. There's extremophiles that have very unique promoter sequences and promoters for certain regions. Question here, is the only way to know which part of a gene circuit is dysfunctional through experimentation? Are there ways to trace the circuit? That's a good question. So again, I'm going to go back to here. This is kind of, if you look here, each one of these you can imagine as an actuator, so as an output, right? And so you could see if you want to test it iteratively, you could. This is actuator design, right? So let's go all the way back to the front. This would be actuator design. So maybe each step of the circuit as you're building it, you can insert maybe a fluorescent protein or something. So there's ways to do it to see how it works and to test it as you go along. But if you want these to be working all in tandem, there's ways to have outputs. You could test, again, you can run Western blots to test to see which protein's being produced or not. Because again, those are all proteins for these more complex systems.
Yeah, you can measure the intermediate protein levels by Western blot, for example.
Yeah. So. And so that's a way to test it. So there's multiple ways. Yeah, you don't have to do it all at once. You can do it all at once or not. Okay. All right, how are these circuits used during wet/lab experiments? So wet lab experiments, you can imagine you just put in the plasmids and hopefully they start working, These plasmids keep going back and forth, sorry y'all. These plasmids were just inserted into breast cancer cells. And it actually worked. They ended up producing these therapeutic molecules. That's wet lab. Dry lab, it's a little bit different. Dry lab, I don't think is really, a lot of its focus kind of even simpler than this, like ligand docking, right? Really understanding that, or understanding how certain, like, I guess, medical molecules would mess around with this stuff. So dry lab's not, I don't think there yet. This is more of a wet lab kind of experiment. Synthetic biology is more wet lab for sure. But that's a great question.
Yeah, but also, don't discount dry lab as well, because, you take what you learn from wet lab and you try to model it in dry lab. And
exactly.
So, a lot of the things that we do here, for example, maybe you want to model HIF alpha levels or TF levels and how that relates to the therapeutic gene output, right? That's dry lab right there. You take, for example, certain binding constants and, all these parameters, you feed it into your computer and it gives you some output. And then now you have a prediction software. So that's kind of cool.
Yeah, and that's it. Yeah, go look at Marcus Covert's whole cell model. That's extreme. That's all modeling, all computational dry lab. Super cool, right? And that's kind of getting towards this, and it's a step in. So a huge goal for bioengineers is to build a cell from scratch. Yeah, exactly. This is like modeling is a step in how you do that because it's just faster. It's cheaper to use computational power rather than doing this in dry lab. And so it's more efficient. But a lot of this DNA like testing and stuff, it's iffy if it can be used or not, because we're still trying to figure out how to model cells in general. Great question.
And there are whole fields, if you want to look more into it, there's a field called pharmacokinetics and pharmacodynamics, PK, PD. These are words that literally, you take data and you try to feed it into a model to predict, hey, if I give you this dose, this will happen. If I give you this dose, this will happen. So you can, if you think about it, that then decreases the risk of bad things happening during clinical trials, for example, because you know how much you need a dose for the desired effect. So modeling is incredibly important, both clinically, environmentally, whatever you really want. Modeling is super important.
I will. Here's some actually, like, I think some of the best advice. Do some CS, do some computer science through your degree. Like, get a computer science minor. I did. It's extremely useful, and you're going to use it if you go into bioengineering. And bioengineering, a lot of students are still a little bit behind on computer science, but for sure, get a computer science like minor at least. Very, very useful stuff. And this would be used for modeling. It'd be used for bioinformatics. It's used for everything. Like you end up doing a ton of coding as bioengineers. So, no problems. Python, yeah, exactly. There's some really cool libraries in Python for bioinformatics. Great question. What are the applications of DNA logic apart from therapeutics / diagnostics? Be creative. Like you can use this stuff for all sorts of things. Me and my friend, who's also a member of BIOME, Ayush, maybe you'll see some of him on some of our videos later if he goes around to making them. But we had a class together, and yeah, we'll see. And we designed something to test for radiation, right? So it's kind of a diagnostic, but this is more real world. And we designed it so it could be on a patch, and so people who work in maybe next to X-rays or something could know if there's radiation or they're being exposed to too much radiation. You can be really creative. You use DNA logic to produce for material science. If you had to produce like spider silk, it's the example I used a little bit ago, or a couple of webinars ago.